My name is Emily Schwarz. I'm a biology major and I work in Jarjour Lab in the Division of Immunology and Rheumatology in the Wexner Medical Center. My project explores the effects of microRNA inhibitors on the immune system in a mouse model of lupus, or SLE. SLE is an autoimmune disease characterized by chronic inflammation and systemic dysregulation of immune function. Our lab has previously shown that toll-like receptors 7 and 8 are significantly upregulated in immune cells in patients with SLE. Toll-like receptors 7 and 8 bind single-stranded RNA to stimulate innate inflammatory responses, but recent studies have discovered that microRNAs packaged and secreted in vesicles can also activate these receptors. We focused on inhibiting 3 of these microRNAs in particular, microRNA 21, 29A, and 29B. Our preliminary data was used to submit a patent for an inhibitor cocktail that blocks these microRNAs. These microRNAs are known to bind either TLR7 or TLR8 and stimulate pro-inflammatory cytokine production in mice and humans. Using a humanized mouse model of lupus, where we take immune cells from SLE patients and engraft them into immunodeficient mice, we were able to examine the effects of microRNA inhibition in vivo. To this end, we isolated immune cells from SLE patients and transfected them with different combinations of microRNA inhibitors or control inhibitor and injected them into the mice. After 3 weeks, we collected blood and measured cell populations by flow cytometry, cytokine production via ELISA, and collected tissue for histology. In examining the cell populations, we found that human cells had successfully engrafted after 3 weeks in both treatment and control groups. The serum cytokines from these mice, however, showed decreased levels of human IL-2, IL-6, IL-10, and TNF-alpha in the microRNA inhibited groups compared to control. Histology also showed robust inflammatory responses in the small intestine, liver, and kidney of the control group and reduced responses in the microRNA inhibitor groups. Immunohistochemistry showed that many of these infiltrates are CD3-positive T cells. Overall, this project helps us understand how the inhibition of microRNAs affects immune signaling and further explores their therapeutic potential in SLE. It would be important for me to participate in the Denman because it would give me the opportunity to both share my findings and develop my communication skills. This is important because my goal is to go to graduate school to get my PhD, and it's valuable to have experience presenting research effectively to be successful in this profession. Thank you.